Imagine you pull a perfectly healthy day 3 embryo out of the incubator, right? You place it on an unheated stage for just for maybe 60 seconds to quickly check its morphology before a transfer.
Right, just super quick look.
Exactly. And it looks completely fine. The blastomeres are intact. There's no obvious fragmentation. But in that one single minute, completely invisible to your eye, the microscopic scaffolding holding its entire genetic future together just shatters.
It is a terrifying reality, isn't it? I mean, you put it back in the incubator, the embryo visually survives the check, but you have just permanently locked in an aneuploidy.
It really is wild. And today, we are exploring exactly why that happens.
Yeah, so for those of you listening, especially you clinical embryologists, deep into your preparation for the ESHRE exam, this is the hidden world we really need to navigate today. Welcome to this deep dive into the cytoskeleton.
I am genuinely thrilled we're tackling this. Our mission today is to completely obliterate the textbook idea of the cytoskeleton as some passive, rigid biological scaffolding, you know?
Oh, absolutely. We have to start seeing it for what it truly is. It's this incredibly volatile, dynamic, force-generating engine that basically dictates every single success or failure in your laboratory.
Because when you think about it, this topic underpins literally every single manipulation you perform as a finch.
Every single one.
Yeah, like when you strip away the branding on your culture media and you look past the calibration of your incubators, what you are fundamentally doing in the IVF lab is managing cytoskeletal mechanics.
That is the perfect way to frame it. From the exact angle you perform ICSI to evaluating a cleavage stage embryo on a time-lapse monitor, right through to the osmotic gradients you use during cryopreservation.
You're constantly directly interacting with these filament systems.
You are. And the clinical stakes are just existential for the embryo. I mean, consider the sheer biological audacity of the first 5 days of human development.
It's a massive marathon. You start with a single, highly specialized gamete, right?
Right? And in just over 100 hours, it must transform into a blastocyst of over 100 cells. It's got differentiated tissues, it's ready for uterine implantation, and every step of that rapid developmental marathon is fundamentally a mechanical hurdle.
Totally. The chromosomes have to be divided with absolute perfect fidelity, and that's a task managed entirely by one specific network.
Yep. Then the massive cell volume has to be physically cleaved. It has to be severed from itself again and again without losing any contents. That's a completely different network.
And before any of that even happens, the oocyte has to parcel out its cytoplasm with this exquisite, deliberate asymmetry.
Oh, the asymmetric division, yes.
It pushes out the polar body, dumping the unwanted chromosomes while hoarding all the metabolic machinery for itself. All the mitochondria, the maternal mRNAs.
And none of which happens without the cytoskeleton generating physical force.
Yeah.
But here's the real clinical sting, and honestly, the reason this is so heavily tested on the ESHRE exam. They're fragile, right? Incredibly fragile. These dynamic structures are not steel beams. They are delicate biochemical polymers, temperature fluctuations, sudden osmotic shifts.
And mechanical shear forces of a biopsy or an injection pipette?
Exactly. They all directly target and disrupt these fragile networks.
I really want to pause and lean into that fragility for a second, because conceptually, I think we often gloss over it in the lab. When we take an oocyte out of its highly controlled environment and we briefly cool it, we aren't just, chilling the cell or slowing down its metabolism for a minute.
No, you are literally melting the M2 spindle.
We absolutely are. If you think about the M2 spindle, it is this highly ordered, dynamic structure holding maternal chromosomes in perfect tense alignment.
And it requires a massive amount of cellular energy and precise chemical gradients to maintain itself. The moment you drop the temperature below 35 degrees Celsius, the biochemical equilibrium just favors disassembly.
So the spindle doesn't just sag a bit, it catastrophically melts. The microtubules depolymerize right back into free protein.
And when the spindle melts, there is nothing holding the chromosomes, so they scatter throughout the cytoplasm.
Which is it's exactly like pulling the foundational pins out of a building while the construction workers are still on the scaffolding. Everything collapses.
And when you warm that oocyte back up, the cell frantically tries to rebuild that spindle and recapture those scattered chromosomes. Sometimes it gets it perfectly right.
But often it gets it wrong. And suddenly you have created the precise conditions for aneuploidy.
It changes the way you look at a plastic dish, honestly. So to understand how to protect these cells, We are going to give you all a comprehensive roadmap today.
We are unpacking 3 distinct interconnected structural systems. We've got actin filaments, intermediate filaments, and microtubules.
But we aren't just going to list their textbook definitions, right? We are going to trace their specialized roles sequentially. We'll start from the formation of the gamete, move through the shock of fertilization and early cleavage, and follow them all the way to the complex architecture of the expanding blastocyst.
Right, because for your clinical practice and your exam prep, you need to understand these structures comparatively. They do not operate in a vacuum.
A cell relies on all 3 systems simultaneously, but for vastly different physical tasks. By the end of this, you really need to have a rock-solid grasp of actin treadmilling, microtubule dynamic instability, and the truly mind-bending process of a centrosomal spindle assembly.
Beyond the molecular mechanics, we'll heavily emphasize 2 critical clinical competencies. First, you must master the asymmetric positioning of the M2 spindle.
Because if you don't understand the physical mechanism of how that spindle gets to the cortex of the oocyte, you cannot understand polar body extrusion.
Exactly, and you won't understand what you are seeing when it fails on the monitor. Second, you must be able to directly link your daily RT procedures to specific forms of cytoskeletal stress.
So you stop being a technician just following a protocol, and you become a true biologist managing a living mechanical system. To build that foundation, let's start by putting these 3 cytoskeletal systems side by side and comparing their base properties.
Okay, let's do it. How do they fundamentally differ in their design?
It is best to organize them mentally by their diameter, the specific monomer they are built from, their polarity, and their dynamics. Let's start with the thinnest and most versatile of the 3 actin filaments.
Also known as microfilaments.
Right. These are microfilaments measuring about 5 to 9 nanometers in diameter. They are built from small globular proteins called G-actin.
And structurally, actin filaments are polar, meaning they have a distinct plus end and a distinct minus end, which is crucial for how they move.
They are highly dynamic. They rely on the hydrolysis of ATP to drive a rapid turnover process called treadmilling. And when they need to generate massive contractile force, they pair up with their specific motor protein, myosin.
Okay, so that's the thinnest. Then in the middle, we have intermediate filaments, or IFs. These measure roughly 8 to 12 nanometers in diameter.
But unlike actin, which is built from those little globular spheres, intermediate filaments are built from long, elongated, fibrous subunit proteins.
And this is the most critical distinction for the exam, right? They are apolar.
Totally apolar. There is no plus end, no minus end. Because of this, they are incredibly stable. They do not require ATP or GTP to assemble, and they have absolutely no motor proteins associated with them.
Which leaves us with the largest of the 3 systems, microtubules. These are massive compared to actin. They have an outer diameter of 23 nanometers.
And unlike the other 2, they form hollow tubes. They are constructed from heterodimers of alpha and beta tubulin. Like actin, microtubules are polar, but they utilize GTP instead of ATP for their energy currency.
And they display a violently active behavior called dynamic instability. They serve as the superhighways of the cell, utilizing kinesin and dynin as their motor proteins.
I think the biggest trap we fall into as embryologists is thinking of these as redundant backup systems. Like, oh, the microtubule network took some damage during a freeze-thaw cycle. Maybe the dense actin cortex can compensate and hold things together.
But they are entirely non-redundant.
Precisely. You cannot compensate for a shattered microtubule spindle with extracortical actin. It just doesn't work that way. They are physically interconnected. They literally link together via cross-linking proteins to form a continuous tensegrity network. But functionally, their jobs are absolute.
You need actin to pinch a cell membrane in half. You need microtubules to drag massive chromosomes across the cell, and you need intermediate filaments to lock those newly formed cells together so they don't drift apart.
Break any single link in that chain, and the embryo fails. This means we really need to understand the molecular how of each system. Let's zoom in on the microfilaments first. Actin.
Okay, I know it's a polymer, but how does it actually assemble from those little globular G-actin monomers?
Think of a mature actin filament as a tightly wound double helical polymer, almost like 2 strings of pearls twisted around each other. The individual pearls are the G-actin monomers.
And each monomer is pretty small, roughly 42 kilodaltons in molecular weight.
Yeah. But a G-actin monomer doesn't just snap into the chain on its own. To be quote-unquote assembly competent, it must be bound to a molecule of ATP.
So the ATP is like the ticket for admission into the polymer chain.
Exactly. The ATP-bound G-actin binds tightly to the growing end of the filament. But once it is physically incorporated into the chain, it actually acts as an enzyme and hydrolyzes its own bound ATP into ADP.
And that biochemical change from ATP to ADP is the critical trigger for movement, right? Because an ADP actin monomer has a slightly different 3D conformation.
Right. It doesn't fit quite as snugly with its neighbors anymore. It's under mechanical strain, kind of itching to break off from the chain, particularly at the older end of the filament.
And this is what establishes the polarity. Actin has a fast-growing barbed end, which is the plus end, where fresh ATP actin is rapidly added. And it has a slow depolymerizing pointed end, the minus end, where old ADP actin is constantly breaking off.
This brings us to treadmilling, which is a concept I always try to visualize as an escalator. The steps of the escalator are continuously moving upward from the bottom floor to the top floor, but the physical structure of the escalator stays the exact same length.
That is a highly accurate visual. In a steady state of treadmilling, the cell is adding new ATP actin monomers to the barbed end at the exact same rate that it is losing older ADP actin monomers from the pointed end.
The filament doesn't get any longer or shorter, but it is physically moving through space.
Wait, conceptually, I've always struggled with that a bit. If the filament is just constantly adding to the front and subtracting from the back, how is it actually generating the physical force to push against something like a cell membrane? It seems like it's just spinning its wheels.
It's a great question, and it really comes down to thermodynamics and physical barriers. Imagine the barbed end of that actin filament is pushed right up against the inner plasma membrane of the cell.
Okay, I'm picturing it.
As thermal energy causes the membrane to fluctuate and microscopic gaps open up for just a fraction of a millisecond, an ATP actin monomer slips into that gap and binds to the barbed end.
Ah. So the newly added monomer acts like a physical wedge.
Exactly like a wedge. It binds, it elongates the filament by just a few nanometers, and when the membrane tries to flex back, it hits the newly extended filament and is physically pushed forward.
That's incredible. Multiply that wedge effect by thousands of actin filaments all treadmilling simultaneously, and you generate massive protrusive force.
That makes perfect sense. But to coordinate thousands of filaments pushing in the right direction, the cell must have a way to control this chaos. The oocyte isn't just a bag of treadmilling actin, it's highly orchestrated.
This is where we get to the regulatory proteins, and for the ESHRE exam, you need to know exactly how this molecular toolkit operates. The first major players are the nucleators.
Because spontaneous actin assembly is way too slow for biological needs, so the cell uses the ARP23 complex.
Right, ARP stands for actin-related protein.
Yes. When the cell needs to build a dense, complex web of actin, like the cortex just under the membrane signaling, molecules activate nucleation-promoting factors like WASP and WAVE.
And those factors grab the ARP23 complex and stick it onto the side of an already existing actin filament.
Exactly. ARP23 acts as a template and initiates a brand new actin filament branching off at a precise 70-degree angle.
So every time ARP23 fires, you get a new branch. That leads to this incredibly dense, interconnected mesh, like a thick thicket of brush. But what if the cell doesn't want a branched thicket? What if it needs a long, straight structural cable to pull against?
Then it completely bypasses ARP23 and uses a different class of nucleators called formins, such as MDA. Formins do not create branches.
They grab G-actin monomers and stack them end to end, acting as a molecular motor that rides along the barbed end.
Right, protecting it from being capped while rapidly elongating a straight unbranched filament.
So ARP23 builds the nets and formins build the cables, but you still have the problem of running out of spare parts. If all the actin is tied up in these filaments, the whole system grinds to a halt.
Which is why the disassembly side of the treadmill is just as tightly regulated. Enter cofilin, also known as actin depolymerizing factor, or ADF. Cofilin specifically recognizes the older ADP-rich actin at the pointed end of the filament.
It binds to it, introduces an agonizing mechanical twist to the helical structure, and physically snaps the filament. It rapidly depolymerizes those old monomers back into the cytoplasm.
But those liberated monomers are bound to ADP, meaning they're useless for building. They need new tickets.
Exactly. That is the job of profilin. Profilin binds to the ADP actin monomer, pries it open, and facilitates the exchange of that dead ADP for a fresh molecule of ATP.
It instantly replenishes the pool of assembly-competent G-actin, ready to be added back to a barbed end.
And finally, you have capping proteins, like CapZ, which I imagine act like the plastic aglets on the end of a shoelace. They bind to the barbed end and just lock it down, completely blocking any further addition or loss.
allowing the cell to freeze an actin filament at a precise length. That is the complete toolkit.
Hmm.
Now let's apply this elegant system to what actually happens in the clinical embryology lab. Let's look at the oocyte cortex.
This is the region right beneath the plasma membrane. It's essentially a dense R23-generated meshwork of actin, and it serves a critical fructural role during our procedures.
Like when you do ICSI, right?
Yeah. When you push an ICSI needle against the zona and the oocyte membrane indents deeply before it finally breaks that resistance you feel, the elasticity of the cell, that is the actin cortex doing its job.
It is the cell's physical armor. But biologically, its primary role is organizational. That actin meshwork is responsible for physically holding the cortical granules in a precise single-file layer right up against the inner membrane.
Which is absolutely vital for the blocked polyspermy. When the sperm enters, calcium spikes, and those cortical granules have to exocytose immediately, dumping their enzymes into the perivitelline space to harden the zona pellucida.
If your actin meshwork is damaged from excessive mechanical stress or suboptimal pH in the media, those granules drift away from the membrane.
And if they drift away, they don't fuse. The zona doesn't harden in time, and your risk of a 3PN fertilization from polyspermy skyrockets.
It is a perfect example of how structural integrity directly dictates fertilization outcomes. But actin's most dramatic feat in the oocyte is undoubtedly spindle positioning.
Oh, the asymmetric division. This is one of the most mechanically fascinating things an oocyte does. The whole point of meiosis in the female gamete is to get rid of half the chromosomes without giving up any of the precious cytoplasm.
But the M2 spindle actually starts out quite centrally located in the oocyte. If the cell cleaved right there, down the middle, you'd get two perfectly equal-sized cells.
Which would be an evolutionary disaster. The resulting embryo wouldn't have enough stored maternal mRNA or mitochondria to survive the journey to the uterus.
So the oocyte has to physically drag that massive spindle from the dead center of the cell all the way to the absolute outer edge, the subcortex. And it does this using an actin highway.
How does it build that highway? It can't just be random treadmilling.
No, it is highly directed. Formins nucleate a dense track of straight actin cables leading from the spindle directly to the near section of the cell membrane.
Once the tracks are laid, motor proteins, specifically myosin V, attach to the spindle poles and literally walk along these actin cables, hauling the M2 spindle outward.
And as it gets close to the edge, the cortex has to catch it, right?
Yes. As it approaches the membrane, the ARP23 complex goes into overdrive. It builds a specialized, incredibly dense cortical actin cap. Think of it like a dense catcher's mitt made of branched actin.
The spindle docks perfectly into this cap, locking it in place right at the periphery, ready for polar body extrusion.
The clinical consequence of this mechanism failing is massive. Let's say a patient's oocytes are subjected to intense oxidative stress, or maybe there was a prolonged temperature drop during retrieval. The actin tracks depolymerize.
The myosin motors have nothing to walk on.
The M2 spindle remains stranded in the center of the cytoplasm.
And what happens when it gets the signal to divide? It attempts a symmetric division. The oocyte literally tries to cleave in half, forming a giant polar body that steals half the cytoplasm.
You end up with a severely cytoplasmically depleted oocyte that, even if it fertilizes normally, simply lacks the metabolic runway to reach the blastocyst stage.
Which perfectly illustrates why dynamic actin is existentially important. But the cell can't survive on dynamic treadmilling alone. As the embryo grows, it needs structures built for pure, unyielding endurance.
So, we must shift our focus to the physical shock absorbers of the cell intermediate filaments.
If actin is a dynamic treadmilling escalator, intermediate filaments are biological climbing ropes. They are incredibly strong, incredibly flexible, and built specifically to resist tension, compression, and shear forces.
They don't have ATP or GTP caps, and they aren't constantly building and breaking down every few minutes.
Their molecular architecture perfectly explains their intense stability. We mentioned they are built from elongated, fibrous proteins, not globular ones.
Two of these long proteins will lay parallel to each other and wind around one another to form a tightly bound coiled dimer.
It's like twisting two strands of yarn together to make a thicker string.
Precisely. Then two of these coiled dimers will line up side by side to form a tetramer. But here is the critical architectural secret: they line up in an anti-parallel fashion, meaning the head of one dimer aligns with the tail of the other dimer.
So because they are facing opposite directions, with equal numbers of protein subunits pointing both ways, the overall tetramer completely loses any sense of directionality. There is no structural difference between the two ends.
Exactly. It becomes completely apolar. There is no plus end to grow from and no minus end to shrink from. These tetramers then stack end to end and bundle laterally to form massive rope-like cables.
But wait, if they have absolutely no polarity and no motor proteins like myosin or kinesin can walk on them, how do things move along them? How does the cell use them for transport?
That is the beauty of it. Things do not move along them. Intermediate filaments are absolutely not transport tracks. The cell uses microtubules and actin as the highways and local roads, but it uses intermediate filaments purely as structural steel.
They anchor the nucleus, they rivet cells together, and they distribute mechanical stress across entire tissues so that individual cells don't tear under pressure.
For the ESHRE exam, you need to know the specific types of intermediate filaments because they are tissue-specific. The type of EF a cell expresses tells you exactly what kind of tissue it belongs to.
Let's run through the major classes. First, the keratins. These are the hallmark of epithelial cells, which becomes extremely important when we discuss the trophectoderm later on.
Then you have vimentin, which is highly expressed in mesenchymal cells and fibroblasts, but notably, it also forms a very loose, flexible cage in early cleavage stage blastomeres.
Then there's desmin, which is specific to muscle cells holding the contractile units together. GFAP, or glial fibrillary acidic protein, which is found in astrocytes in the nervous system.
Neurofilaments, which act as the internal rebar for long neuronal axons.
And finally, the lamins, specifically lamins AC and B1, B2. Lamins are structurally unique because they are the only intermediate filaments located inside the nucleus rather than in the cytoplasm.
They form the nuclear lamina, a dense, cross-linked meshwork just inside the inner nuclear envelope that protects the shape of the nucleus and organizes chromatin.
For embryologists, lamin B3 is a standout. This is the oocyte-specific isoform. An oocyte sits in meiotic arrest for decades. It needs incredible nuclear stability during that time, and lamin B3 provides that protective cage around the maternal DNA.
So how do these massive cables actually integrate with the rest of the cell to form cohesive tissues? Because an early embryo eventually has to stick together tightly to become a blastocyst.
They don't just float freely in the cytoplasm. Intermediate filaments form a continuous mechanical network stretching from the nuclear envelope all the way to the plasma membrane.
We just mentioned the paranuclear cage protecting the nucleus. Massive cross-linking proteins like plectin physically reach out from that cage and bolt the intermediate filaments directly to the actin cortex and to the microtubule network.
It creates a unified tensegrity system. If you pull on the actin membrane on one side of the cell, the force is transmitted through the IFs directly to the nucleus and out the other side. But the real structural magic happens at the cell junctions. This is how individual cells rivet themselves together to form an embryo.
We need to talk about desmosomes. These are the heavy-duty cell-to-cell junctions. On the extracellular side, spanning the gap between two neighboring cells, you have these specialized cadherin proteins, desmoglein and desmocolin. They reach across the space and lock together like teeth in a zipper.
But a zipper is useless if it's sewn into weak fabric. So on the intracellular side, right under the membrane, you have adapter proteins like plecofilin and desmoplakin.
These adapters act like heavy-duty steel brackets. They grab the tails of the cadherins and anchor them directly into the massive keratin intermediate filament network inside the cell.
The mechanical result of this is profound. When trophectoderm cells are linked by desmosomes, they stop acting like individual cells. They become a mechanically continuous sheet.
If fluid pressure pushes against one trophectoderm cell, the force isn't borne by that cell's membrane alone. It is instantly distributed along the keratin networks of the entire epithelium. Hemide desmosomes do the exact same thing, but instead of linking cell to cell, they anchor the base of the cell to the underlying basement membrane using integrins.
There is a vital developmental nuance to this, though. If intermediate filaments make cells so stiff, resilient, and locked together, why do early blastomeres, like a 4-cell or 8-cell embryo, express incredibly low levels of intermediate filaments?
It seems counterintuitive. You'd think the embryo would want to be as strong as possible right from the start. But the key is plasticity. Those early blastomeres are undergoing rapid, dramatic cleavage divisions. They are completely changing their shape and internal volume every 12 to 24 hours.
If a 4-cell embryo was densely packed with rigid keratin networks and locked together by massive desmosomes, it physically couldn't cleave properly. The contractile ring wouldn't be strong enough to pinch through the keratin steel.
So the embryo deliberately suppresses strong IF expression, relying instead on a very loose, flexible Vementin cage, remaining highly plastic until it is absolutely ready to compact and differentiate.
The timing is perfectly regulated. It waits until day 3 to turn on the structural steel. So we've covered the dynamic actin treadmills and the stable IF ropes. Now we have to contrast all of that intense stability with the most volatile, explosive, and frankly dangerous structures in the entire cell, microtubules.
Let's dive deep into their structure and this bizarre phenomenon of dynamic instability.
Microtubules are massive structural tubes. At 23 nanometers in outer diameter, they dwarf actin filaments. Structurally, they are hollow cylinders built from exactly 13 parallel protofilaments arranged in a circle.
And each of those protofilaments is a long chain of repeating subunits, an alpha-tubulin tightly bound to a beta-tubulin. This alpha-beta heterodimer is the fundamental building block.
And because they always stack in the same orientation, alpha, beta, with beta-tubulin always exposed at one end, the entire microtubule is polar.
The end exposing the beta-tubulin is the plus end. This is the end that reaches outward toward the cell periphery and experiences rapid growth and shrinkage. The alpha-tubulin end is the minus end, which is typically firmly anchored deep in the center of the cell.
But unlike actin, which uses ATP to drive its treadmilling, tubulin relies entirely on GTP.
And this GTP dependence is the secret to dynamic instability. I want to spend some time visualizing this because it is heavily tested and conceptually tricky. When a free alpha-beta tubulin dimer is floating in the cytoplasm, the beta-tubulin subunit is holding onto a molecule of GTP.
As long as it has that GTP, it is primed and ready to be added to the plus end of a growing microtubule.
As the cell rapidly adds these fresh GTP tubulin dimers to the plus end, it builds what we call a GTP cap. You can think of the GTP cap as a molecular clamp. The inherent physical shape of a tubulin dimer actually wants to curve outward.
But when it is bound to GTP, that outward curvature is suppressed. The GTP forces the dimer to remain perfectly straight, allowing the 13 protofilaments to pack tightly together into a stable tube.
I always picture it like a massive, tightly coiled steel spring that is under immense tension, held together at the very tip by a tiny, fragile plastic clip, the GTP cap. As long as you keep adding new plastic clips to the top faster than they can break, the spring stays straight and the microtubule grows.
That is a fantastic analogy, because the critical factor here is time. Shortly after a tubulin dimer is incorporated into the tube, it naturally hydrolyzes its GTP into GDP. So the body of the microtubule behind the cap is composed entirely of GDP tubulin.
And GDP tubulin desperately wants to curve outward.
It's under immense mechanical strain. The only thing keeping the whole tube from ripping itself apart is that tiny protective layer of GTP tubulin at the very tip, the GTP cap.
So what happens if the concentration of free tubulin in the cytoplasm drops slightly, or a temperature shift slows down the addition of new dimers? The rate of adding new GTP tubulin slows down, but the relentless wave of hydrolysis behind it keeps going.
Eventually, the hydrolysis catches up to the very tip.
The plastic clip dissolves. The GDP cap is lost.
And the result is violent. The microtubule undergoes what is literally termed a catastrophe. Without the GTP cap forcing them straight, the 13 protofilaments violently peel outward, curling back on themselves like a banana peeling in fast motion.
The microtubule rapidly depolymerizes, shrinking many times faster than it originally grew, scattering GDP tubulin back into the cytoplasm.
It shrinks and shrinks until, purely by chance, it might suddenly bind a fresh cluster of GTP tubulin, reestablishing a cap. This starts the catastrophe and it starts growing again. This recovery is called a rescue.
So, pushing back on the evolutionary logic here, why on earth would a cell rely on a primary structural component that is a ticking time bomb? It spontaneously self-destructs if it slows down for even a second. It seems incredibly inefficient and dangerous.
It sounds like terrible biological design, I know.
Yeah.
But if you look at the physical problem the cell is trying to solve, dynamic instability is a stroke of pure genius. It is a highly efficient search and capture mechanism.
When a cell is preparing to divide, it has to find and physically attach to tiny little protein patches, kinetochores, located on chromosomes that are randomly scattered throughout a massive volume of cytoplasm.
The cell has no eyes. It can't aim a microtubule directly at a chromosome.
Exactly. So it relies on rapid, blind exploration. It nucleates hundreds of microtubules in all random directions. They shoot outward, exploring the 3D space of the cell. If a microtubule shoots out into empty space and finds nothing, it loses its GTP cap, undergoes catastrophe, and rapidly shrinks.
This instantly reclaims all those tubulin monomers so they can be used to try a different direction. It doesn't waste resources building a bridge to nowhere.
But if a microtubule randomly bumps into a kinetochore, boom! The kinetochore chemically grabs the plus end, it stabilizes it, it prevents the loss of the GTP cap, that specific microtubule is captured, it stops shrinking, and it establishes a permanent physical tether to the chromosome.
The cell just blindly explores its entire volume until every single chromosome is captured.
And clinically, the pharmacology of IVF heavily leverages our understanding of this exact instability. We use drugs to manipulate the GTP cap. For instance, Taxol, or paclitaxel, binds directly to the side of a microtubule and completely stabilizes it, preventing depolymerization even if the GTP cap is lost.
Which sounds like a good thing, making it stronger, but it's actually fatal to the cell. A functional meiotic spindle must be dynamic. The microtubules need to constantly shrink and grow to generate the tension required to align chromosomes. If you freeze them in place with Taxol, the spindle is paralyzed and division fails.
Conversely, we use drugs like colchicine, vinblastine, and nocitazole in the lab to study these structures. These drugs bind to free tubulin dimers in the cytoplasm and prevent them from polymerizing.
Without the ability to add new dimers, the GDP cap is inevitably lost, catastrophe ensues, and because no new tubulin can be added, the entire spindle completely melts away.
Okay, so if these dynamically unstable tubes are constantly shooting out, exploring space, and collapsing, where do they actually originate from? They can't just spontaneously coalesce out of nowhere in the cytoplasm, right? That would be total chaos.
You're right. Spontaneous nucleation from free alpha-beta tubulin requires a massive local concentration and is far too slow and disorganized for a living cell. Instead, cells use a dedicated, preformed template called the gamma-tubulin ring complex, or gamma-2RC.
Gamma-tubulin is distinct from the alpha and beta subunits.
Yes, it doesn't form the main tube. The gamma-2RC is a massive protein complex that contains exactly 13 gamma-tubulin monomers arranged in a perfect, rigid circle. It perfectly matches the 13 protofilament architecture of a mature microtubule.
It's essentially a pre-made molecular starter ring. The cell lays down this gamma-2RC where it wants a microtubule to begin. The free alpha and beta tubulin dimers see this perfect ring and just start rapidly stacking on top of it.
This gamma-2RC firmly nucleates and anchors the minus end of the microtubule, protecting it from depolymerization, while allowing the plus end to shoot outward into the cytoplasm to do its searching. In a normal somatic cell, thousands of these gamma-2RCs are densely concentrated in a central organelle called the centrosome, specifically embedded in a fuzzy cloud of protein called the pericentriolar material, or PCM.
So the centrosome acts as the master organizing hub, anchoring all the minus ends in the center of the cell, with all the plus ends radiating outward like spokes on a bicycle wheel. And once established, these microtubule spokes become the primary highway system for the cell.
And they have specific, highly directional motor proteins that run on them. Kinesins are plus-end directed motors. They walk anterograde, meaning they grab vesicles, organelles, and mRNAs and haul them from the center of the cell out toward the periphery.
While dyneins are minus-end directed motors, they walk retrograde, grabbing cargo at the cell membrane and pulling it back into the center of the cell toward the centrosome hub. A brilliant clinical example of this highway in action is in the blastocyst.
Yes, the expansion of the blastocle.
Trophectoderm cells have to pump massive amounts of water into the center of the embryo to expand that cavity. To do this, they need aquaporin water channels inserted into their apical membranes. They package these aquaporins into vesicles and use kinesin motors walking specifically along microtubule tracts to deliver those vesicles precisely to the apical surface.
If your microtubule network is compromised, the aquaporins never reach the membrane, water isn't pumped, and blastocle expansion halts.
It is all mechanically driven, but here is where we take a dramatic, almost paradoxical turn in the biology of early development. We just established that the centrosome is the central, indispensable hub. It is the master organizer of microtubules in a normal dividing cell.
But what is utterly fascinating and crucial for embryologists to understand is that the human oocyte completely abandons this fundamental rule of biology.
Yes. The plot twist, the meiotic spindle in the mammalian oocyte is entirely acentrosomal. During early oogenesis, long before the follicle even matures, the oocyte deliberately destroys its own centrioles.
It literally throws away the master instruction manual and the physical hubs for building a spindle.
And yet, despite lacking a centrosome, it still has to assemble a massive bipolar meiotic spindle capable of segregating chromosomes with enough precision to create a healthy human being. So, how does it build the M2 spindle without a centrosome?
It is one of the most perilous mechanisms in biology.
Mhm.
It uses two complementary pathways that operate simultaneously, and both are incredibly precarious. Let's look at the first one, the inside-out pathway, where the chromosomes themselves drive the nucleation.
This relies on a small GT paste called RAN. The chromatin of the chromosomes possesses an enzyme that maintains a highly localized, dense clyde of RAN GTP directly surrounding the DNA. As you move away from the chromosomes into the cytoplasm, this concentration drops off sharply, creating a chemical gradient.
This high concentration of RAN-GTP right next to the DNA acts as a spatial signal. It activates specialized spindle assembly factors, proteins like TPX2 and NUMA, which are floating nearby.
Exactly. When TPX2 is activated by RAN-GTP, it triggers the nucleation of microtubules directly at the surface of the chromatin. So instead of microtubules shooting out from a centrosome to find a chromosome, the microtubules literally start growing outward from the chromosomes themselves.
That's the inside-out pathway. But that just gives you a messy sunburst of microtubules around the DNA. You need to organize them into two distinct poles to pull things apart. This is where the second outside-in pathway comes in.
Scattered loosely throughout the oocyte cytoplasm are hundreds of tiny, disorganized clusters called MTOCs, or microtubule organizing centers. These contain the gametubulin and pericentrin we discussed earlier, but they aren't structured into a central centrosome.
They are essentially loose, floating puzzle pieces. As the chromosomes nucleate their own microtubules outward, these floating MTOCs are dragged toward the DNA by motor proteins like dynin. They begin to coalesce and cluster together, forcing all the minus ends of the newly formed microtubules to converge into two focused points.
And those two focus points become the bipolar ends of the M2 spindle. This raises a monumental point about clinical vulnerability. Because this acentrosomal assembly is inherently slower, more chaotic, and entirely reliant on delicate, invisible gradients of chemical factors floating in a vast cytoplasm, it is intensely sensitive to environmental disruption.
This is the exact reason why oocytes are so much more fragile than cleavage-stage embryos. A cleavage-stage embryo has inherited the sperm centriole during fertilization. It has a real centrosome to build a robust mitotic spindle.
But an unfertilized oocyte is relying on this precarious RAN-GTP gradient. A slight drop in temperature on your bench, and that delicate balance of MTOC clustering and RAN-GTP signaling shatters. The spindle melts, and it struggles to reassemble perfectly without a central hub to guide it.
Thankfully, the oocyte does possess a powerful biochemical safety mechanism to prevent it from dividing while the spindle is a mess, the spindle assembly checkpoint, or the SAC. This is a crucial concept for ESHRE prep.
The SAC is a cell's internal quality control inspector. Specifically, it involves proteins like MAD1 and MAD2, which physically localize to the unattached kinetochores on the chromosomes. As long as even a single kinetochore is not perfectly attached to a microtubule, these MAD proteins scream a biochemical SOP signal into the cytoplasm.
That SOP signal actively inhibits a massive protein complex called the anaphase promoting complex, or APCC. The APCC is the master switch that triggers cell division. So as long as MAD1 and MAD2 are firing, the APCC is shut off, and the cell is physically locked in metaphase arrest, unable to divide unless every single chromosome is captured.
However, we know as clinicians that this checkpoint is absolutely not foolproof. Embryos missegregate chromosomes all the time. So let's dive into exactly how chromosomes are supposed to segregate, how they are held together, and why advancing maternal age is so incredibly devastating to this specific process, because this is the root molecular cause of aneuploidy in the IVF clinic.
To segregate correctly, a chromosome must achieve what is called an amphitelic attachment. This means it is perfectly bioriented. One kinetochore is firmly attached to microtubules pulling toward the left pole, and the sister kinetochore is firmly attached to microtubules pulling toward the right pole.
This creates a physical tug-of-war. The kinetochores are being pulled in opposite directions, which generates immense mechanical tension across the center of the chromosome. The cell physically measures this tension to verify that the attachment is correct.
But things go wrong. If the attachment is syntelic, meaning both kinetochores accidentally attached to microtubules coming from the same pole, there is no tug-of-war. The chromosome will just be dragged whole to one side. Or worse, a meritelic attachment, where a single kinetochore is simultaneously snagged by microtubules from both poles. It gets stuck in the middle and often tears.
When these erroneous attachments occur, there is a lack of proper tension. The cell senses this lack of tension using an error-correcting enzyme called Aurora B kinase. Aurora B localizes right between the kinetochores. When tension is low, the kinetochores are physically close to Aurora B, and the kinase phosphorylates the attachment proteins, actively destabilizing and severing the microtubule connection. It forces the microtubule to let go so it can try again.
It is a continuous cycle of trial, error, severing, and reattachment until perfect amphitelic tension is achieved. Okay, so let's say all attachments are perfect, tension is established, the SAC is satisfied, and stops sending the SOP signal. How does the cell actually release the chromosomes so they can fly apart?
They don't just drift apart. They are physically welded together by massive protein rings called cohesin. Cohesin encircles the sister chromatids, locking them tightly together. To separate them, you have to cut the rings.
When the SAC is finally satisfied, the anaphase promoting complex, the APCC, is activated. The APCC acts as a targeted assassin. It tags a specific inhibitory protein called securin for immediate destruction by the proteasome.
Securin's entire job is to hold another enzyme hostage. That enzyme is separase. When the APCC destroys securin, separase is suddenly unleashed into the cytoplasm, and separase acts like molecular scissors. It zeroes in on the cohesin rings, cleaves them in half, and in a single synchronized moment, the chromosomes snap apart and are dragged to the poles by the microtubules.
But there is a crucial, heavily tested distinction here regarding the two stages of meiosis. In M1, the first meiotic division, you are separating homologous chromosomes, the maternal and paternal pairs. But you absolutely need the two sister chromatids of each chromosome to stay tightly glued together for the subsequent M2 division.
Right. If separase just blindly cuts all the cohesin during M1, the sister chromatids would fall apart prematurely and M2 would be a chaotic disaster. So how does the oocyte selectively cut the cohesin on the arms of the chromosome, but leave the cohesin at the very center, the pericentromeric region, perfectly intact?
It uses a brilliantly named protector protein, shugoshin. Shugoshin literally translates from Japanese as guardian spirit. During M1, shugoshin localizes specifically and exclusively to the pericentromeric region of the chromosomes. It acts as a shield. It recruits a phosphatase enzyme that continuously removes any activating phosphates from the cohesin in that specific area.
By doing that, shugoshin physically prevents separase from recognizing or cutting the cohesin at the center. So during M1, separase only cleaves the cohesin on the distal arms, allowing the homologs to separate. The guardian spirit ensures the pericentromeric cohesin isn't cleaved until the oocyte is finally fertilized and completes M2.
Which brings us to the profound tragedy of reproductive aging, that cohesin. The vital protein glue holding those chromatids perfectly together so the kinetochores face outward to be captured. It is loaded onto the maternal DNA when the oocyte is first formed in the fetal ovary, before the patient is even born, and it is never replaced or replenished.
Decades pass. A woman reaches her late 30s or early 40s. That exact same cohesin ring has been sitting in that oocyte, bathed in reactive oxygen species, for 40 years. It slowly degrades. The rings spontaneously break and fall off.
The physical consequence is devastating. Without tight cohesin holding them firmly together, the sister chromatids become loose, they flop around. The kinetochores, which should be rigid and facing perfectly opposite directions, begin to wobble. Because they wobble, the microtubules make constant, erroneous syntelic and meritelic attachments.
And Aurora B kinase just can't keep up with the sheer volume of errors. It tries to correct them, but the geometry of the chromosome is too distorted. To make matters exponentially worse, the SAC proteins themselves, MAD1 and MAD2, transcriptionally decline in abundance as the oocyte ages. So the safety checkpoint itself becomes leaky. It stops shouting SDOP even when errors are present.
The clinical statistics are brutal, but they make complete molecular sense once you understand cohesin depletion. In oocytes retrieved from women under the age of 35, the aneuploidy rate is roughly 20 to 25%. But in women over 40, as cohesin fails universally across the cohort, that aneuploidy rate skyrockets to 50 to 70%. It is a direct, unavoidable physical readout of cytoskeletal and molecular decay over time.
It really reframes how we counsel patients regarding age. It's not just egg quality as some vague concept. It is the physical degradation of microscopic protein rings. But let's assume the chromosomes do separate correctly. The next monumental task is to physically cleave the massive volume of the cell in half. Enter cytokinesis, and the contractile ring.
This is such a stunning display of localized mechanical force. The chromosomes have been segregated to opposite poles by the microtubules. Now, the cell needs to know exactly where the equator is so it can pinch precisely in the middle and ensure both sets of DNA end up in separate cells.
It uses the remnants of the microtubule spindle to find the center. The microtubules left behind in the middle of the cell, the central spindle, recruit a master regulatory protein called ECT2. ECT2 localizes directly to the equatorial plasma membrane.
ECT2 acts as a localized activation switch. It activates a small GTPase called Rho-OA, but crucially, it only activates Rho-OA in a tight, narrow ring around the exact equator of the cell. Nowhere else. Active Rho-OA is the master commander of the contractile ring. It simultaneously fires off two distinct signaling pathways.
First, active Rho-OA recruits and activates a formin protein. And we know from earlier that formins rapidly nucleate long, unbranched, straight actin cables. So suddenly you have a massive burst of actin cable formation wrapping in a ring around the cell equator.
Second, Rho-OA activates an enzyme called ROCK. ROCK stands for Rho-associated coil kinase. ROCK's primary job is to target the regulatory light chain of myosin II. It phosphorylates the myosin, causing its tail domains to uncoil and assemble into active, bipolar, thick filaments.
So perfectly aligned at the equator, you now have a fresh track of parallel actin filaments, and you have hyperactivated aggressive myosin motors binding to them.
They form the actin-myosin contractile ring. It is incredibly precise and remarkably thin, only measuring about 0.2 micrometers wide. As the myosin motors begin to walk furiously along the actin filaments, they generate intense sliding force. The ring physically constricts, like pulling a purse string incredibly tight, dragging the plasma membrane inward to form the cleavage furrow.
It pulls and pulls, invaginating the membrane until there is only a microscopic, dense bridge of cytoplasm left connecting the two new cells. This bridge is called the midbody.
But the contractile ring alone cannot make the final cut. It can squeeze, but it cannot fuse the opposing membranes to seal the cells. For that, a specialized protein complex called ESHRE spirals into the midbody tube and literally snips that final membrane tether. That precise final cut is called abscission, and it physically completes cytokinesis.
What's deeply fascinating here is how the oocyte modifies this exact standard mechanism for polar body extrusion. Polar body extrusion utilizes the exact same molecular machinery, ECT2, Rho-OA, formin, ROCK, myosin, but it executes it as an extreme, highly polarized version of asymmetric cytokinesis.
Exactly. I love your water balloon analogy for this.
It's the only way I can visualize the volume difference. If you have a massive water balloon and you tie a string exactly around the middle and pull it tight, you end up with two perfectly equal halves. That's a normal symmetric cleavage division. But polar body extrusion is like grabbing just the tiny absolute tip of the balloon's rubber, pinching it off, and tying the knot right there at the edge. You save almost all the water in the main balloon and discard just a tiny, useless knot of rubber containing the extra chromosomes.
On a molecular level, the contractile ring forms strictly over that subcortical M2 spindle that the actin tracts dragged to the edge earlier. The Rho-OA signal is restricted just to that tiny cortical cap. It constricts and pinches off an area that represents maybe 1/20th or even less of the total oocyte volume.
And when this goes wrong clinically, if the contractile ring fails to constrict fully because of actin disruption, or if the spindle wasn't perfectly docked at the edge, you don't get a clean polar body. You get a retained polar body. Or you trigger massive, chaotic oocyte fragmentation right at the membrane as the machinery tries and fails to pinch off.
So we've successfully managed the cytoskeleton to fertilize the egg and cleave it a few times. We have a day 3 embryo. Let's follow this embryo as it develops further. The cells are going to start specializing and building tissues, which means we need to bring intermediate filament strongly back into the narrative.
Right. Let's trace the timeline. Pre-compaction, from the 2-cell to the 8-cell stage, the embryo intentionally keeps its structural intermediate filaments very low. We established this. It uses that loose fermenting cage for plasticity to allow for rapid cleavage. But at day 3, usually around the 8 to 16 cell stage, compaction occurs. The embryo transforms from a loose cluster of distinct grapes into a smooth, tightly bound sphere, and everything changes mechanically.
At compaction, the cells radically alter their surface adhesion. E-cadherin catenin complexes, which are transmembrane adhesion proteins, suddenly engage the dense cortical actin networks lying just beneath the plasma membrane of each blastomere.
This physical linkage cadherin to actin literally pulls the adjacent cells tight against one another, maximizing their contact area and flattening them out.
As they physically flatten and press together, The blastomers positioned on the outside of the embryo begin to rapidly accumulate keratin intermediate filaments. These newly minted keratin cables immediately wire themselves directly into the heavy-duty desmosomes that are forming between the outer cells.
This is the critical transition. Suddenly, that outer layer of cells isn't just a group of individual blastomers touching each other. They are mechanically welded together by a continuous keratin desmosome network. They become a true epithelial sheet, the presumptive trophectoderm.
And to make that sheet functional, they seal the uppermost gaps between the cells with tight junctions, utilizing sealing proteins like claudins and ZO1, which also tie back into the actin cytoskeleton. This sealing is biologically imperative because by day 5 or 6, to become a true blastocyst, the trophectoderm has to start moving fluid.
The Na-PPIC plus ATPase pumps on the basolateral membrane start aggressively moving sodium ions into the central space of the embryo. Water follows the sodium osmotically to expand and pressurize the blastical cavity. If those tight junctions aren't perfectly sealed by the tension of actin and keratins, the water just leaks right back out into the culture media, and the blastocyst collapses or fails to expand.
Notice, however, how the inner cell mass, the ICM, behaves completely differently during this time. The cells trapped on the inside remain relatively disorganized. They maintain very low desmosomal coupling, low keratin expression, and high plasticity. They've absolutely not committed to being a tough, rigid outer shield.
Which brings us to one of the most profound, paradigm-shifting concepts in modern developmental biology, and a massive point for advanced exams. We always think of genetics driving structure, but here, the physical cytoskeleton doesn't just shape these cells, it actually dictates their genetic destiny.
Yes, the mechanical switch for lineage specification. It is a stunning example of mechanotransduction, where a cell translates a purely physical force into a biochemical gene expression profile.
This still blows my mind every time I review it. Let's unpack the physics. We have a compacting morula. The cells on the outside are being physically stretched thin to cover the entire surface of the embryo. To resist that stretching and maintain their shape, their actin-myosin cortex goes into overdrive. That intense actin-myosin contractility generates high cortical tension on the membrane.
That high mechanical tension is the physical signal. The cell possesses mechanosensors at the membrane that detect this tightness. In these tense outer cells, the physical strain causes two specific co-activator proteins, known as YAP and TAZ, to translocate straight from the cytoplasm into the nucleus.
YAP and TAZ cross the nuclear envelope. And once inside the nucleus, they bind to DNA and drive the massive expression of a gene called CDX2. And CDX2 is the master transcription factor that permanently locks a cell into becoming trophectoderm. High tension equals CDX2 expression.
But look at the cells trapped deep inside the morula. They're squished by the outer layer. They are not stretching to cover a surface. Therefore, they have very low cortical tension.
Because they lack that physical tension, a completely different signaling cascade, called the hippo pathway, becomes highly active in the cytoplasm.
The active hippo pathway unleashes a kinase cascade that ultimately phosphorylates YAP and TAZ. When YAP is phosphorylated, it is physically trapped in the cytoplasm. It cannot cross the nuclear envelope.
Without YAP in the nucleus, CDX2 is never turned on. The trophectoderm program remains completely silenced. Instead, those relaxed inner cells default to expressing OCT4 and NANOG, and OCT4 and NANOG are the core definitive pluripotency factors. Those low-tension cells become the inner cell mass, the future fetus.
So if we connect this mechanotransduction back to the bigger picture of the IVF laboratory mechanical tension, is the upstream determinant of cell fate. It is not just chemical gradients diffusing through the embryo, it is sheer physics.
Which means our laboratory conditions can directly interfere with genetics. If you are using a suboptimal culture media that slightly alters osmolality or causes the blastomeres to swell or shrink abnormally, you are literally altering the cortical tension of those cells.
Which means you could accidentally, mechanically misallocate cells. A cell that is positioned on the inside, which was supposed to become ICM, gets osmotically swollen and stretched too tightly. It senses erroneously high tension, sends YAP to the nucleus, turns on CDX2, and accidentally differentiates into trophectoderm. You have fundamentally altered the lineage specification of the embryo just by altering the physical environment of the dish.
That is a terrifying segue into our next vital topic, RE procedures and cytoskeletal vulnerability. We are constantly handling these cells. Let's talk about the myriad ways a well-intentioned embryologist can accidentally disrupt these exact mechanisms, starting with the most ubiquitous variable, temperature.
We covered this briefly in the introduction, but we must revisit it with our new deep molecular context. The M2 spindle depolymerizes completely below 35 degrees Celsius. When you cool an oocyte by taking it out of the incubator onto an unheated surface, you strip the GTP caps off the microtubules. Catastrophe ensues, the protofilaments peel back, and the spindle vanishes, leaving the chromosomes to scatter in the cytoplasm.
The crucial clinical question is how quickly does this happen, and is it reversible? If I take a dish out for just 60 seconds to check an alignment on an unheated stage, does the spindle just snap back perfectly together when I put it back in the warm incubator?
It is a gamble, and this is a vital concept for the ESHRE exam. The ability to reversibly reassemble the spindle depends heavily, almost entirely, on the biological age and quality of the oocyte. In a young, robust oocyte from a 25-year-old donor, with massive reserves of free tubulin, highly active gametoses, and plenty of intact cohesin rings holding the chromatids perfectly tight, the spindle might reassemble and recapture the chromosomes perfectly upon rewarming.
But what about an oocyte from a 41-year-old patient?
In that older oocyte, the cohesin is already severely degraded. The chromatids are loose and wobbling, the spindle assembly factors are depleted. When you melt the spindle with a cold shock, the chromosomes scatter. And when you rewarm it and the MTOCs try to desperately rebuild the spindle from the RAN-GTP gradient, those loose, wobbling kinetotors are prime targets for meritelic or syntelic attachments.
And because Aurora B kinase is already overwhelmed and MAD2 is declining, the cell fails to correct the errors. The damage from that brief 60-second cold exposure is now locked in as a permanent aneuploidy during the next division.
That is why strict thermal control is non-negotiable. What about cryopreservation? Freezing and thawing oocytes and embryos.
Here, the primary enemy is osmotic flux, which exerts extreme mechanical shear force on the cytoskeleton. When you load an embryo into high-concentration cryoprotectants, massive amounts of water rush out of the cell. The cell membrane shrinks dramatically and aggressively. When you warm it and dilute the cryoprotectants, water rushes back in and the cell swells rapidly.
This rapid cycle of shrinking and swelling puts immense physical tearing stress on the dense actin cortex and the fragile microtubule networks. If the cell shrinks too fast, the rigid actin cortex can physically fracture, leading to blebbing or lysis. This is exactly why the stepwise equilibration protocols moving embryos carefully through increasing and decreasing concentrations of CPAs are so absolutely vital. You must strictly manage the rate of osmotic change to give the dynamic actin treadmill time to remodel and adapt to the shrinking membrane without tearing.
And then there's ICSI. This is perhaps the most direct, aggressive mechanical intervention we perform. We take a rigid glass needle, push it deep into the cell, and physically break the plasma membrane to deposit a sperm. That is a massive mechanical shear stress that violently tears the cortical actin network at the exact site of injection.
Which is why operator technique is the defining variable in ICSI survival. You want a clean, swift, highly controlled entry. But more importantly, you must completely avoid excessive cytoplasm aspiration. Some embryologists aggressively suck massive amounts of cytoplasm into the injection needle to ensure the membrane is broken.
When you do that, you aren't just moving fluid. The cytoplasm isn't water. It is a dense, cross-linked gel of intermediate filaments and microtubules. When you aspirate aggressively, you are literally tearing the actin meshwork, violently displacing organelles, and ripping the intermediate filament networks away from their nuclear anchors. You can physically destroy the architecture of the cell.
Let's also touch on pharmacology in the lab, because sometimes we intentionally chemically disrupt the cytoskeleton to make our jobs easier. Cytoskeleton B and cytoskeleton D are potent actin polymerization inhibitors. They bind to the barked plus ends of actin filaments and prevent the addition of new ATP actin, halting treadmilling.
We use them during specific biopsy procedures, right? Like during polar body biopsy or older zona drilling techniques. Because cytoskeleton temporarily softens the dense, rigid actin cortex, making the cell highly pliable and making the physical biopsy much easier to perform without shattering the blastomere membrane.
It is highly effective for that, but there is a dangerous physiological trade-off. If you expose the oocyte or embryo to cytoskeleton for too long, you don't just soften the cortex, you begin to melt the critical actin tracts nucleated by formins that are holding the M2 spindle locked at the extreme edge of the cell.
So without the actin tracts holding it, the M2 spindle begins to drift back toward the center of the oocyte. And if you activate the oocyte while the spindle is drifting centrally, you risk a catastrophic symmetric division and a massive loss of cytoplasm.
Precisely. Okay, we have talked extensively about how invisible, vital, and fragile these structures are. But how do we actually see them and assess their competence in the clinical lab without destroying the cell? We need to cover cytoskeletal biomarkers and clinical assessment tools.
This is where advanced optics come into play. We do have tools to visualize this biology directly. The pulscope, or spindle rotation imaging, is a phenomenal technology. It utilizes polarized light microscopy to read the birefringent retardants of the M2 spindle.
Because the microtubules in the spindle are so perfectly aligned, parallel, and densely packed with protein, they literally shift the phase of polarized light as it passes through them.
The brighter and more distinct the signal on the pulscope, the more highly ordered, dense, and robust the microtubule spindle is. If the signal is weak, fuzzy, or completely absent, it tells you immediately that the spindle has depolymerized due to thermal stress, or is highly disorganized due to age-related cohesin failure. And practically, we use this to guide ICSI.
Exactly. You map the exact location of the spindle relative to the first polar body. We assume the polar body marks the location of the spindle. But polar bodies drift in the paravitaline space over time, so looking at it under standard bright field isn't enough.
With the pulscope, you find exactly where the true M2 spindle is docked, and you ensure you inject the sperm needle precisely 180 degrees away from it, or at least at a safe angle. This ensures your glass needle doesn't physically plow into and shatter the microtubule array. Also, if you notice a severely misaligned spindle relative to the polar body, it is a strong biological warning sign of aberrant cortical positioning and potential actin dysfunction.
We also harvest a massive amount of cytoskeletal data passively through time-lapse monitors and morphokinetics. When you are annotating an embryo, every strange abnormal division pattern you record is a direct visual readout of a specific cytoskeletal failure. For example, direct unequal cleavage, or DUC, where a single blastomere attempts to divide directly from one cell into 3 distinct cells simultaneously.
Direct unequal cleavage is a catastrophic, total failure of cytokinesis. It usually means the cell failed to form a bipolar spindle and instead formed a tripolar microtubule spindle. Or the ECT2-ROA mechanism misfired and the actin contractile ring failed to localize into a single, clean equatorial band, instead trying to pinch the cell in multiple directions at once. DUC is a massive red flag that is heavily associated with complex aneuploidy.
What about multinucleation at the 2-cell stage? You evaluate a 2-cell embryo, and inside one blastomere, you see 2 distinct pronuclear-like structures.
That means the microtubule spindle likely did its job. The chromosomes separated and formed 2 nuclei, but the actin-myosin contractile ring completely failed to constrict and sever the membrane. It's a failure of cytokinesis, trapping 2 nuclei in one shared cytoplasm. Or consider excessive fragmentation. When you see those small nucleate fragments bubbling rapidly off the surface of a cleaving cell, you are looking at direct evidence of localized actin contractile ring dysfunction, where the membrane is pinching off chaotically rather than in a single controlled furrow.
So when you sit in a dark room annotating a time-lapse video, you aren't just clicking buttons to log developmental milestones. You are directly grading the molecular competence of that specific embryo's actin and tubulin networks.
Okay, we have covered an immense, almost overwhelming amount of ground. Let's bring all these threads together for a final, rapid-fire review of the key concepts you absolutely must have locked down for the ESHRE exam.
Let's recapitulate the core principles. You must definitively know the structural distinctions of the 3 systems. Actin is 5 to 9 nanometers, utilizes ATP for treadmilling, nucleates via R23 or formins, and physically powers the contractile ring and cortical dynamics. Intermediate filaments are 8 to 12 nanometers, apolar, incredibly stable, require no energy, and provide immense tissue strength via desmosomes and keratins. Microtubules are massive 23-nanometer hollow tubes, utilize GTP, display violent dynamic instability, and form the meiotic and mitotic spindles to move chromosomes.
You absolutely need to understand the mechanism of actin treadmilling and precisely how myosin VB motors and ARP23 cortical caps physically drag the M2 spindle asymmetrically to the subcortex to allow for proper, highly polarized polar body extrusion.
You must grasp the nuance of microtubule dynamic instability, specifically the protective function of the GTP cap, and how the mammalian oocyte uniquely relies on a precarious acentrosomal RAN-GTP gradient-driven spindle assembly.
Remember the intricate mechanics of chromosome segregation. Aurora B kinase acts as the tension sensor to correct erroneous attachments. Cohesin degrades irrecoverably with advancing maternal age, leading directly to wobbly kinetochores, failed SAC checkpoints, and those terrifying aneuploidy statistics 50 to 70% in women over 40.
For physical division, trace the signaling pathway. ECT2 activates ROA strictly at the equator, which subsequently activates both formin and ROCK, assembling the ultra-thin actin-myosin contractile ring to squeeze and sever the cell.
And for the developing blastocyst, remember the mechanotransduction switch. High cortical tension on the outer cells drives YAP and TAZ into the nucleus, activating CDX2 and locking in trophectoderm lineage. Conversely, low tension on the inner cells activates the hippo pathway, trapping YAP in the cytoplasm, allowing OCT4 and NENOG to specify the inner cell mass.
I fully acknowledge this is a staggering volumes of mechanistic detail to absorb. But the unifying foundational principle here is remarkably simple for clinical practice. Cytoskeletal integrity is the ultimate metric of laboratory success. If you can maintain strict environmental controls to protect these fragile filament systems, you protect the embryo.
Which brings us to the end of our deep dive. Thank you for joining us on this incredibly dense, fascinating journey into the cellular mechanics of early human development. In our next session, we will be looking at specialized cellular structures, which build directly on the architecture we've discussed today.
But before we sign off, I want to leave you with a final forward-looking thought, something that pushes beyond the established science we've discussed today and points to the very near future of clinical embryology.
I love looking ahead. Where is this field going next?
We spent this time talking about how cytoskeletal errors, like a wobbly kinetochore or a delayed contractile ring, lead directly to aneuploidy. Currently, the only way we definitively test for that aneuploidy is by physically tearing off a piece of the trophectoderm for PGTA. But the cytoskeleton generates movement. It generates distinct physical signatures.
Right, the speed of cleavage, the exact duration of the contractile ring pinching.
Exactly. And right now, researchers are training advanced artificial intelligence models to analyze high-frequency time-lapse morphokinetics. The AI isn't just looking at the timing of cell division, it is analyzing the microscopic subcellular movements of the membrane, the cytoskeletal micromovements that are entirely invisible to the human eye.
Wait, are you saying the AI can literally watch how the actin cortex flexes or how the spindle aligns and use that physical movement profile to predict the genetic status of the embryo?
That is exactly where the technology is heading. Because the movement is driven by the proteins, and the proteins are dictated by the genetics and structural integrity we've discussed, a chaotic aneuploid division creates a distinct chaotic micromovement signature on the membrane. In the near future, we may be able to perfectly predict ploidy non-invasively simply by having an AI assess the mechanical competence of the actin and microtubule networks through the camera lens. The cytoskeleton won't just be the engine of the embryo, it'll be the ultimate non-invasive diagnostic readout. Keep that in mind the next time you're evaluating an embryo on the screen.
Thank you sincerely for your patient listening and dedication to mastering clinical embryology. It's professionals like you who advance our field every day. If this content added value to your practice, a simple like helps it reach more embryologists and assisted reproductive technology specialists worldwide. Please subscribe and turn on notifications so you never miss updates on lab protocols, cryopreservation tips, and guideline-driven insights. Your support fuels more free, high-quality resources for our entire assisted reproduction community. Let's grow this channel together for better patient outcomes.